Hello, biology students. This is going to be the beginning of chapter 11. The chapter is going to be focused on DNA. And in the last chapter, we talked about how genetics is going to be inherited from the parents to the offspring. In this chapter, we're going to be looking at DNA and its structure and function a little bit. So with that being said, there are a couple sections here. And the first section is going to be about the history of DNA and how we discovered what DNA was. So there's a lot of information here that you don't have to write down, which is a good thing. I like to still go over it just to kind of talk about how do we come to these conclusions about DNA. And I think it's interesting how these scientists kind of came to their conclusions. So early 1900s, again, this is after Mendel discovered his Mendelian genetics. Scientists were still kind of confused about how traits were passed from parent to offspring. We knew about cells, we didn't know about all the structures within the cells, but we started to figure out that there was some substance in the nucleus that's going to be really important for determining the characteristics of a cell and an organism. And you can kind of guess that's already DNA. We didn't know what DNA looked like. We didn't know what DNA, how it functioned. But basically, we started to understand that DNA was somehow involved in the cell producing its features. So then, once we did figure out that it was DNA, we started trying to figure out what the structure or how it looked and what it was made up of. And because scientists realized that this was such a profound molecule that the first person to figure out the structure of it was going to be known forever. So Rosalind Franklin was a scientist during that time in the 1930s and 40s who had a PhD in science. And remember, this time period, there wasn't a lot of females in science, but Rosalind Franklin was one of the top scientists that was using something called X-ray crystallography. And she was actually looking at how that could be used to figure out the structure of DNA. Now, she took photos of the DNA, and these photos were long exposure. So how you have your phone, your camera, you can take a photo in like a millisecond. These pictures usually took hours and sometimes even days to develop, so it was a lot longer of a process. But basically what she did was she used this machine called an X-ray crystallography machine to take a picture called Photo 51. And that was clear evidence of the structure of DNA, which is a double helix. It's this twisted ladder. What happened was one of Franklin's coworkers stole her picture and showed it to two other scientists that were trying to figure out the structure of DNA. And they used this data to build the first model of DNA. Watson, Crick, and Wilkins were awarded the Nobel Prize for their discovery because they published their research, not really first, but the journal that they published in made it seem like they discovered it first, and they just saw, or they put Franklin's research behind kind of supporting theirs. And Franklin actually never was awarded the Nobel Prize because she sadly passed away before the actual presentation of the medals. So and you can't win Nobel Prizes after somebody has died. So it wasn't really known that Rosalind Franklin's data was the actual data that led to Watson and Crick developing their first model. And they actually were kind of mean in a book they wrote about the discovery. They called her Rosie, which she didn't like. So they were kind of really just mean to her and stole her data and didn't give her credit. So I like to use this time to show that students that this is I call it a black eye of science. It's not a good memory that we have, and it's really important to kind of highlight the contribution that Rosalind Franklin gave to the discovery of DNA, because without her, Watson and Crick wouldn't have gotten to their discovery. So it was really Rosalind Franklin that helped us discover the structure of DNA. Hello, everyone. We're going to go over the second section in chapter 11, and this is going to be more about DNA. Again, this whole chapter is going to be focused on DNA. In this specific section, we're going to be talking about the nucleotides that make up DNA. If you remember back in our chapter on biochemistry, we talked about how DNA is a polymer while the monomer is a nucleic acid. So think like bricks making a wall. Nucleic acids are made of a monomer known as a nucleotide. So we're going to be looking at how these nucleotides are arranged in DNA. Remember that DNA is the blueprint of all your proteins, or blueprint for yourself, and it just makes you who you are. So again, DNA is made of nucleotides. They're the monomer. They're the building blocks of DNA, which is a polymer. That is a big concept that we have to understand. How these nucleotides are going to be bonded together is going to be really the main focus of this chapter. So, nucleotides. there are three parts. There's a phosphate group. You can see this is that phosphate group where we have a phosphorus element, an atom, surrounded by 4 atoms of oxygen. Sugar molecules, deoxyribose. If you think ribose, that ending ose is a sugar. That's one way to remember it. So this is that sugar molecule. We also have a nitrogen base here. It can be a single ringed structure or a double ring structure. If you're looking here, this is a double ring structure because there's 2 rings. Now, there are 4 different types of nucleotides. There are in DNA. There is adenine, there is guanine, there is cytosine and thymine. If we look here, adenine and guanine are going to have 2 rings, while cytosine and thymine only have 1. But these are our 4 different types of nucleotides. Now, between each nucleotide, the sugar and the phosphate group are going to be identical. So no matter what we're looking at, whether it's adenine and guanine or cytosine and thymine, thymine and adenine, each of the phosphates and sugars are going to be exactly the same. What makes them different is whether or not they're a double or single ring structure. So we can see here we have adenine, which is a double ring structure. Thymine and cytosine are single ring, and guanine here is a double ring structure. So again, we just count the number of rings to figure out which one it is. Pyrimidines are the single ring structures. I remember as pyrimidine, the Y. If you think cytosine and thymine, they have Y's in their names. That's one way to remember it. Purines are a double ring structure. Adenine and guanine are purines because they have 2 rings. They're a double ring. You can see here, each purine is going to bind specifically with a pyrimidine. So adenine is going to bond with thymine. Cytosine is going to bond with guanine. Guanine will never bind with thymine, and adenine will never bind with cytosine. So exactly what I just said there, each purine is going to bond with a specific pyrimidine. You can see adenine bonds with thymine, guanine bonds with cytosine. I like to think apples in the trees, cars in the garage, or these 2 are curved, these 2 are straight. So I like to show this with my students. You can see here, adenine bonds with thymine, guanine bonds with cytosine, cytosine bonds with guanine, thymine bonds with adenine, so on and so forth. Good little meme here. I'm sorry, G, but we're basically not a good match. Think of this as being these are bases, and they are not going to match. They're not going to bind together. They're not going to bond together. Adenine will only bind with thymine. Guanine will only bind with cytosine. Last, we have something called the Chargaff rule. This is the percent of adenine is going to equal the percent of thymine in DNA, and the percent of guanine is going to equal the percent of cytosine. This is because, again, they bond with each other. Now, there is a small problem set that we have to kind of understand. If we understand that, like, thymine and adenine are going to be the same, guanine and cytosine are going to be the same, if you find a bacteria species and the percent of thymine is 12, what's the percent of adenine? Well, this is easy. It's 12 as well because they're the same. But if you look at, the percent of cytosine, which is 43%, what's the percent of guanine? It's also 43%. So these problems are pretty easy. However, when we get to the opposite, so we think about thymine is 10%. What's the percent of cytosine? What we have to do is we have to take thymine and realize, okay, well, same as adenine, so that's 20%. So the adenine thymine percent is 20%. If we take that minus 100, we're going to get 80. So that's going to be our Cg concentration. So we just got to divide that by 2 to figure out what the other 2 would be. Here's another way of looking at it. Again, 34% for guanine. What is adenine? Well, we add those 2 together. We subtract it from 100. We find 32 is the adenine thymine concentration. And then lastly, A and T would be 16. However, when we're going to go over section 3 of chapter 11, this is going to be about the structure of DNA. Specifically in the last chapter, we went over nucleotides. Remember, nucleotides are the monomer for DNA. So these are the components of the structure of DNA. And we're going to see how those nucleotides are going to bond together to form that structure. So DNA consists of 2 chains of nucleotides and a twisted ladder. So you can see this is that twisted ladder. It looks like a ladder with a big twist. This is called the double helix, and this is what Rosalind Franklin discovered back in the 1940s and 50s. These rungs, or the places where you place your hands on a ladder, these are made of nucleotides. So you can see here we have each nucleotide being represented by a sugar, a phosphate, and a base. So you can see this is what the structure of DNA looks like at a larger level. Now, on the outside where this green area is, these nucleotides are held together by covalent bonds. And if you remember back from our chapter on chemistry, covalent bonds are really strong. And that should make sense to you because the backbone of DNA needs to be really strong. Think of your backbone. A weak backbone, you have many problems. A strong backbone, you don't have that many problems. Same with DNA. We need a strong backbone, so it's going to be made of covalent bonds. We call this backbone the sugar phosphate backbone. These are the outsides of the DNA, so you can see here that both sides have this sugar phosphate backbone, and they basically support the nitrogen bases on the inside. Now, these nitrogen bases are also bonded together through something called hydrogen bonds, and if you remember back again from our chapter on chemistry, these hydrogen bonds are going to be kind of weak. There are 3 hydrogen bonds between guanine and cytosine and only 2 between adenine and thymine. That's why they pair together. So adenine won't bind with cytosine because it won't form these nice hydrogen bonds. And this should also make sense to you because hydrogen bonds are weak because we need to read the DNA. We need to be able to split this up and actually read what that sequence of DNA is. These 2 sides of DNA are going to run anti-parallel to each other. So you can see here, these are going to run in opposite directions. I think of like a highway where you travel in 2 directions. So one car goes one way, the other car goes in the opposite direction. They're driving parallel, but anti-parallel because they're going in opposite directions. Now, with this 3 and 5 prime end, we label the end that starts with the sugar as a 3 prime end, and the end that starts with a phosphate group a 5 prime end. That's because of the bonding of the sugar and phosphate. You don't need to know that, but just remember 3 prime is the sugar, 5 prime is the phosphate. And if you look, it's the opposite in the opposite direction. So you can see it's 5 prime on this side, 3 prime here, 5 prime here, and 3 prime here, because they run in opposite directions. Our next section is going to be about replication, which is going to be the copying of DNA. We've talked a little bit about replication before. This is going to be the process that copies DNA, but this is where we're going to end for section 3. All right, let's go over section 11.4, which is going to be about DNA replication. Again, this is the process of copying DNA. And it produces an exact copy of DNA. And if we remember back when we talked about mitosis, this is because each new cell needs a copy of that exact DNA. So again, what we're going to see is a strand of DNA being separated, and it's going to be copied on both sides. That's going to give us 2 identical daughter strands of DNA. Now, this occurs during S phase of the interphase portion of the cell cycle. So again, we have G1S, G2 during interphase. These are the parts of the cell cycle that kind of prep the cell to go through mitosis. During S phase, this is when replication is going to occur. And this is, again, S stands for synthesis. So we're making new DNA. During replication, each new strand of DNA, or each old strand, sorry, is going to be used to make a new strand of DNA. It's going to be used as the template. So I love this image when looking at DNA replication. You can see DNA is going to be split. The new copy here is red. Then it's going to be split again. The new copy is this time green. It's going to be split again. The new copy here is black. So you can see each time the old strand or each side of the strand is going to be used as a template. And we call this being semi-conservative, because one end or one side is conserved while the other is new. So you can see again, we split up, we make new, we split up, we make new, and we split up, and we make it new. So I like to end on this meme because, yes, this is what DNA replication looks like after it's done. We have 2 exact copies of DNA. All right, so this is going to be a little bit more about DNA replication. This is where it does get a little bit more complex, but DNA replication starts at somewhere called the origin of replication. This is just a region on the DNA where DNA is going to start being replicated. So you can see here, this is that replication origin or origin of replication. You can see as it unfolds here, it's going to start replicating, or it's going to start being copied. What happens is helicase is going to come in. It's actually going to unzip the DNA. It's going to open up that DNA to have those template strands be used to make new DNA. So this helicase enzyme, again, enzymes and usually an ACE, are going to break those hydrogen bonds. You can see what that looks like here. This is helicase. Here's another just kind of image of what helicase looks like it does. You can see it just splits open that DNA and allows it to be copied. What happens then is we have something called the replication fork. And this replication fork is just where the DNA helicase is going to unzip the DNA. So right here is the helicase, and it's just opening up the strand of DNA. Here's my analogy, is that this is the origin of replication. This is where you're always going to start. As you're reading, that is like the replication fork, because you're moving where you're reading So wherever this mouse is, this is where the replication fork is. The origin of replication is different. This is where it actually begins. This story begins right here. This is where the origin of replication would be. Where you're reading is the actual replication fork. Next in our section here, we're going to talk about an enzyme called DNA polymerase. This DNA polymerase is going to be an enzyme that's going to bind free-floating nucleotides to the growing chain of DNA. So what it actually does, as you can see here, this DNA polymerase is going to basically just make the other side of DNA. It's going to do that just by binding free-floating nucleotides, the nucleotides we talked about, that are located in the nucleus, and it's just going to make the other strand of DNA. That's all it does. Now, the important thing about DNA polymerase is it only travels in a 5 to 3' direction. So what it does is it makes in a specific direction, kind of like your car drives on the road. There's only one way that it should drive, but this DNA polymerase is going to drive on one side. So what it does is, again, it just makes new DNA. Because it makes new DNA in one direction and DNA is anti-parallel, we have two sides of this strand. We have the leading strand and we have the lagging strand. The leading strand is going to be made continuous. So you can see it's made towards the replication fork, but DNA polymerase can't move in that same direction on the other side. It has to go in the opposite direction. So as it opens up, what's going to happen is new sections of DNA are going to be copied, and those are going to be called Okazaki fragments. This is what it looks like. You can see here the DNA is going to be opened up. The DNA polymerase is only going to be able to move in one direction. So what happens is it's going to be made continuous. That's the leading strand. The bottom strand here is the lagging strand because it's made discontinuous. It's made in little chunks called Okazaki fragments. But you can see the end goal or the end result is still two strands of DNA. Again, this is the replication that we're talking about. Top strand's the leading strand. It's going to be made towards the replication fork. The bottom strand's the lagging strand. It's made away. It's making these Okazaki fragments. Now, our end product of DNA replication is just two identical strands of DNA. That's it. It's making DNA because, again, we need a copy of that DNA for the new cells when they are created. This is the end of chapter 11.